I welcome Professor Amit Sethi. This is Smita Wangikar, sir.
Hello, good
Pres-
afternoon.
yeah. So Professor Amit Sethi is a faculty in electrical engineering, IIT Bombay, and his basically area of interest is deep learning, machine learning, image processing, and computer vision, and even medical image analysis. So, related to medical image analysis, today he is going to talk about modeling survival in prostate cancer using pathology image and R. So, basically, he uses R for his research. Is that correct, Professor Amit?
Yes, thank you.
Yeah. So, yeah, over to you, sir.
Okay, thank you. So, good afternoon, everyone. As Smita mentioned, I work in computational pathology, which means that I work very closely with pathologists. Together, we analyze cancer tissue slides. So, these glass slides that you would see in the microscope, instead of looking at it manually, it will be scanned by a digital scanner. So the scanner has an inbuilt microscope. So it will scan it at very high resolution. So it will map half a micron by half a micron or quarter micron by quarter micron of tissue area to 1 pixel of the image. So you get a very large image. So you have cameras that give you megapixels today. These are gigapixel images. Uh, and then we develop algorithms based on computer vision and image processing and deep learning to extract features from them so that we can make more standardized diagnosis of cancers and also go beyond just automating the work of a pathologist. So pathologists would give a grade to the cancer, that is grade 1, grade 2, grade 3. In prostate cancer there is Gleason grade, which is based on whether you see the cells forming rings of a gland or not. So grade 3, 4, and 5 are the cancer grades in prostate cancer. But within grade 3, if you just do surgery, there can be differences in outcomes. Some people may have cancer come back, some people may not have that recurrence of cancer. So question is, can we predict that? And if we predict that someone is at a high risk of recurrence, then can we escalate their treatment so that they don't have to suffer the coming back of cancer? So let me share my screen. So this is a slide from Beck et al.'s work from 2011. So this is basically just one of the first studies where what they did was they looked at these digitized slides, and based on that they extracted features from these slides, and then they fitted a machine learning model to predict which person will is likely to survive longer and which one is not likely to survive longer. So basically, what we are doing is we are looking at pathology in a whole different way. So if you have a glass slide, usually from that data we would make the diagnosis, including grading and staging, and based on that we will plan the treatment. And after the treatment is done, if we observe the person for a few years, then we will know whether the person has survived or not, whether the cancer came back or not, etc. So this middle part where on which the treatment is based, a lot of that is subjective. And even though there are guidelines on how to grade cancer, etc., there are differences even among expert pathologists. Because there are questions such as, did we see 1% of cells that look like this? So to estimate that 1% among millions of cells, it's just done by approximation. Plus there are places where we don't have expert pathologists, and sometimes they actually make mistakes, and it's not just some subjective disagreement. On the other hand, there are subtle changes in tissue that cannot be seen or cannot be observed very reliably. So the whole idea is that instead of going through this middle, so start from the fact and then end up in a fact, in between use an opinion as a bridge, why don't we go, sorry, directly from fact to fact? So why don't we take the tissue, and if we know the patients for 5 years or so, and we know the outcome, so pick the tissue that was there 5 years earlier and predict what, try to build a model for predicting what happened 5 years later. and then we can use it on future patients to plan the treatments better. So we can decide who is likely to have a non-aggressive cancer so that they don't need to be given a very aggressive treatment, and who has an aggressive cancer for which some side effects can be tolerated but harsher treatment is necessary. So basically, what we do for that is we use machine learning, and basically what machine learning does is, let's say we have different features that we are extracting from cancer. Let's say we are looking at the radius of the cancer nuclei and the size of the gland, and basically all the blue dots are those that did not have a bad outcome, in the sense that after surgery the cancer did not come back. And let's say these are the red dots are the ones where the cancer came back after surgery. So machine learning finds these boundaries to classify these individual patients, so that we can make this prediction for future patients. Now you can see that no machine learning model is fully accurate, and you will see that even with the best efforts, sometimes you will have misclassifications, such as these blue dots in the red areas and the red dots in the blue areas. So that is to be expected, but can we minimize it, and can we make it general enough so that it applies to data from new patients? And now this is, I can only show you plots for two features. We are actually doing this for several features, sometimes thousands of features. Okay, so we are not only looking at the nucleus radius, we are looking at variance in the radius, we are looking at uh, shape of the nuclei, we are looking at eccentricity of the nuclei, etc. So it can keep going that way. Uh, we are trying to actually go beyond what a human can do. We are not just mimicking an expert. We are trying to predict outcomes, not just grades, right? And the decisions can be based on many variables. If we ask an expert to make a decision, usually after 5 to 7 variables, it's very difficult to make decisions on more variables. But a machine can do it very reliably, and the decisions will not be subjective. There will not be any interperson variation, and it can be based on very subtle features that are very difficult for us to quantify as humans, and it can be based on many more examples. A pathologist in their entire career would only see, let's say, a few hundred cases or a few thousand cases. Here you can train a machine to work with 10,000 cases, 100,000 cases, so that it will amass the knowledge of so much. uh, I would caution to basically say that this does not mean that machines will replace pathologists. It just means that they will become a tool that pathologists should also try. In the sense that machine learning works only when you pose very specific problems. If if you if the problem is a little more general, then the machine doesn't do very well. So basically, what happens in prostate cancer screening is that you have uh, when the when the cells lose their structure and they stop forming glands, then usually the cancer forms. And when that happens, then the cells secrete something called prostate specific antigen in the bloodstream. And if that is elevated, and it is found to be elevated in a test, then what happens is that a biopsy is done where a needle is stuck into the prostate, some tissue is taken out, and slides are prepared, and those slides are examined under microscope. Now, the data that we were given is this CPCTR data. It is a collaborative effort between several institutes in the US to collect this data. And what they did was they gave us matched data. That means they gave us data in two groups. Those group in which the cancer came back after surgery, and another group in which the cancer did not come back after surgery. Now, if you see carefully, here you see there's a ring that is forming, one ring and then another ring and then all these other rings. So these rings are glands. And when these ring type structures are there, then it is a low grade cancer. When it becomes more chaotic and rings are merging into each other, then it becomes a medium grade cancer. And when you see no rings at all, then it becomes a high grade cancer. So this grade is indicative of how dangerous the cancer is, but it does not tell us the complete picture. So this is grade 3 when you see these circles. So you can see that this one and this one both are grade 3, yet there are differences in the sense that in one of them the cancer comes back after surgery, in the other it doesn't come back after surgery. So they gave us a matched cohort where for every grade 3 person in the recurrent group there was a grade 3 person in the non-recurrent group. For every grade 4 person there was a grade 4 person. So there was matching between the two groups based on grade, stage, race, and age. Uh, so this is the uh, pipeline that we developed. These are different pieces of the pipeline, stages of the pipeline. The first part of the pipeline was that we looked at this tissue image and we annotated. We took a digital pen and we annotated where the cancer is. So after annotation, we did all these other steps such as process image, export features, prepare data, classify, interpret. I'll go through these in quick succession. So once these images are annotated, so then we can basically get the regions that have the cancer. And after that, we started processing the images. So that means from this image, whichever region had cancer, we built a computer vision pipeline that divided the entire area into different regions that it can recognize. So if you see this dark purple region, that is the gland region. That is this gland region. And if you see this light chewing gum type pink region, so that is basically a computer marking this region, which is this connective tissue, the stroma. And when you see the blue and the black dots, those are all the nuclei. So these are cell nuclei that you see. There are some nuclei that are in the stroma. There are other nuclei that are in the glands. The one in the glands are the ones that are of interest to us because the cancer starts in the gland. And within the gland, there is this opening, the white opening that you see. It's called the lumen. And this is where the gland secretes whatever juices it produces. In this, it is the prostatic fluid or the seminal fluid. And this when you have high grade cancer, we will not see lumen, and the same is the case in breast cancer, for example. Any questions so far? Okay, so to mark all these regions, we use a software called Definitions Developer. That software itself is quite expensive. So this was done in 2014-15. After that, we have started building our own uh, computer vision software to do this recognition of different parts of the tissue. But back then, basically, it looked at different image features, and based on that, it classified each area as one of these. Either it is epithelium or stroma or something which is cannot be resolved between epithelium and stroma, whether it is inflammation or lumen or stuff inside the lumen, which is luminal debris, and then or lymphocytes. These are the white blood cells, or nuclei, or some sort of a clearing in the nucleus and dark nuclei. So once we have that, the other part of the image processing was to classify between epithelium and stroma. And the way we did that was we did that part outside the Definitions developer interface. So this is the interface that I wanted to show from that software. That epistroma classifier, we developed that in R. So what we did was we divided the image into small segments, and for each segment we extracted a lot of features. So such as what is the average redness, what is the average blueness, and so forth, and what are some of the texture features for using computation that is called gray level co-occurrence matrix, GLCM. So all these we applied to extract multiple features from different segments of the tissue. And once we had all those segments, we used a classifier. We used R to build a classifier. And then the results of the classification were uploaded back into this program. And based on that, we were able to get these uh, classification of where the stroma is and where the epithelium or the gland is. So I will show you how we do it in R in a bit, but I'll first give you the complete picture and then I will show you how it is done in R. All right. So part of this epistroma classifier was that we actually had too many features that the software could extract from the image and its segments. So we need to fit something called a regularized model, L1 regularized or lasso regularized model to eliminate a lot of these features that are not used. So we basically looked at so we fitted a logistic regression model where we tightened the penalty slowly in steps. I will show you how that is done in R. And once we did that, based on if we make it too tight, then a lot of the features, their coefficients become zero. So when their coefficients become zero, then they stop that they fall out of the model and they are not useful at all after that. And we did that until our validation error started to increase. And when it started to increase, at that time we stopped that. So that is the most parsimonious or the most compact model that we could get without compromising on validation error. And this is an example of the kind of features that it extracts. So it looks at the mean redness, mean of a particular stain, which is one of the colors, and then it looks at these texture features like GLCM and GLDV, and a blue to red ratio, and a bias term. So I will show these later once I show you the R interface. So once we did that, and we were able to mark all the epithelium and stromal regions and all the nuclear regions. After that, we exported a lot of features. So we exported for each image around 6,000 features. So examples of these features are, for example, how many, if we have the epithelial region, what is the average blue to red ratio of that region. So basically we are modeling the color characteristics of that the epithelial region. And so I think it's better to show something like yeah, so these are like all different variables. In the stromal matrix, what is the color? In the cytoplasm, what is the color, etc. And what is the shape of the nuclei? What are the size of the nuclei? Also some relational features. How are nuclei are of one type, how far away are they from nuclei of another type, and so forth. So if we have 6,000 features and we have only 400 patients or 300 patients, then we have a high dimension low sample size problem, and it is very easy to overfit on this data because there are a lot of features and not too many patients. So we have to eliminate a lot of features. So we look at their correlation with each other. And so this is basically the correlation matrix, color coded. So the diagonal is high correlation as required. But whenever you see off diagonal red element, that means this feature and the feature in the column are very highly correlated. So one of them can be eliminated, for example. And then we would do multiple rounds of that regularized logistic regression where we tighten the regularization penalty and it eliminates more features. So ultimately, once we have that, so from around 3,000 features, we brought it down to either 74 or 18. So these are two different models, one with 74 features, the other with 18 features. So once we have these different, so let's take the example of 18 feature model. So once we have that, we will use those features to model cancer recurrence versus non-recurrence. And to model that, we want to look at both. We ideally want both low false positive rate and low false negative rate. So what that means is that if there is recurrence, then it should call it as recurrence. If there is non-recurrence, it should call it as non-recurrence. If we just look at only one of these two, then we can compromise on the other two, on the other one. So to basically look at both of these together, we look at something called ROC curve, where we plot the sensitivity of the model and 1 minus the specificity of the model. So specificity goes from 0 to 1 in a right to left manner, and sensitivity goes from 0 to 1 in a bottom to top manner, just as expected, but specificity is reverse. Or you can think of it as if this was 0 and this was 1, then this would be 1 minus specificity. So this diagonal line that you see, if a curve comes out to be on the diagonal line, that means the model is not predictive at all. It doesn't do very good. It is just gives you chance prediction. The curve should be above the line, and the further above the line it is, so ideal curve would go straight up here and then straight right here. So that means it is very accurate. So here we got, and then the area under this curve will be 1 if it is like perfectly going straight up and straight right. uh, Anything that is below that, we will get area that is less than 1. The higher the area, the better the model. If the curve is on this line, the area will be half, which means the model is not useful at all. And if it is below that line, that means the model actually predicts opposite of what it should predict. So these are some of the features that came out to be important in that feature elimination process. So for example, and they are not very easily interpretable, but slightly more interpretable than if we were to use a deep neural network. So for example, we got nucleus of epithelium. What is its max relative area to the hole within the nucleus itself? And what is the skewness of layer 3, which is RGB, blue layer, um, and so forth. So they are quite esoteric. It's because we wanted to extract a lot of features and see which ones work better, and some of these turned out to be not so interpretable. Some of them are interpretable. For example, lumen standard deviation of border index. So border index is the smoothness of the gland, and if there is high standard deviation, that means the person is more likely to have is less likely to have cancer come back. Uh, so we are looking at now these features, and now we are trying to interpret it, that what does it actually mean in terms of physical shape in the cancer. So what you can see is that if we're looking at this the mean difference in stain 2, for example, we can see that for cancers that recur, the nuclei become lighter, whereas here they remain darker. Okay, so this could be an indication for things for a pathologist to look at. So we are discovering features that they should look at. And before that, we do something else, which is called color normalization, because tissue from different labs looks slightly different. So sometimes we have to make this tissue look like this tissue without changing the structure. So you can see that we are able to do that. This now looks as pink as this one and less red like this one, but without changing the structure. And for that, we use a mathematical concept called non-negative matrix factorization, which we use to separate out the two stains, the purple and the pink stain. And then we use the same structure that we had, but the color target we used from the other image. Now let's look at the R code. Before we go there, are there any questions?
Sir?
Yes.
Oh. Sir, I wanted to ask about sir, how the sensitivity was related to simplicity, and like you said that the line of the sensitivity will go straight and then will become horizontal, which means that the prediction is perfect. So, sir, like
Yeah. So if all the positive and negative samples can be perfectly separated, then this line will go straight up and then straight right.
okay.
So that means there's a gap between there's a clear gap in which you can draw a line to separate the positive versus recurrence positive versus recurrence negative images. Every time we misclassify, you will see this change, this slight bump to the right, or yeah, so slight bump to the right you will see early in the curve. So because of that, the area under the curve will go below 1.
Hello.
Yes.
Sir, I want to know how you are reducing the attributes.
Yeah, so I will show you how we are doing that. So there is something called lasso regression, lasso logistic regression,
Okay.
where so if you look at the weights of all the features, right? So let's say you have 10 features and you have weights W1 to W10. Now, if you add all their absolute values, then that becomes the L1 norm of the weights. Okay, so we put a penalty on the L1 norm of the weights. So the optimization tries to reduce the L1 norm of the weights. Instead, if we did L2 norm, we will be adding the squares of the weights. Instead of squares, we are adding their absolute value. The advantage of adding absolute values as opposed to squares and trying to reduce the sum of absolute values is that that can only happen if it sets some of the weights to zero, which doesn't happen for squares. Because in squares, when it tries to reduce them, it will make the weights very small. And because square of small is much smaller, so it stops reducing them to zero. It just keeps them as very small numbers. Whereas we want exact zero so that we can eliminate those features. I want to see if I can increase the size of this. Is my screen legible or is it too small? Okay, let me copy paste this in somewhere else so that I can show you it in a bigger manner. Okay, so now this should be visible, right? So this is the R code that I picked up from that R Studio. So this is R Studio. Is everyone familiar with R Studio?
Yes, sir.
Okay, great. So this R Studio that I'm using is part of Anaconda. I use it as part of Anaconda because I also use a lot of Python, and Python gives me both a Jupyter Notebook interface for Python as well as RStudio for R. Uh, Whereas you can also get RStudio just as a standalone. You don't need Anaconda for that if you're not working in Python. So here, this particular example, I'm trying to fit a logistic regression classifier. And for that logistic regression classifier, I have that L1 penalty, the penalty on the sum of absolute values, and that the proportion of the penalty I will slowly increase until I get the desired I drop the desired number of variables, and my accuracy is still high, and I get a small enough model. So basically, this read CSV is to read the training data, another read CSV is to read the test data, and this is the path for the path for the file that I have to read. Right after that, the column that I'm trying to predict is the first column, and this is the penalty. So the penalty is going up in steps from 0 to 29. Okay, 0 to 29 times 0.25. That means every time it is going up by a factor of 0.25. So it goes from 0 to 0.25 to 0.5 to 0.75 to 1 in 30 steps, 0 to 29. Okay, so this is log of lambda. I will take e to the power of this. And then the model type is whether it is logistic regression or SVM. You can select that using model type. And I will do 9-fold cross validation. That means I will divide the data into 9 different segments, fit the model on 8 segments and try it on the 9th one, and I will rotate which one I keep out so that I don't get a fluke model. I test it on the entire data. So then I'm reading the first column as the label, and everything that is not the first column minus C or dependent column as the independent variable. I do the same for the test data. And then I'm using some libraries, liblinear. So liblinear is a library to fit these linear models such as linear regression, logistic regression, ridge or L2 penalized linear regression, L1 penalized linear regression, etc. And then there are some other libraries like Caret and ML Bench. These help me create different folds for the data so that I can fit on k-1 folds and test on the kth fold. And PROC helps me produce that ROC curve that I showed, whose area is supposed to be below 1, but ideal area is 1. Okay, so I take all these libraries and then I am running a loop over all these log lambda. So in 30 steps, the one that I showed you above. And within that, I will first train the model on everything except one fold. And then I will predict it on the test fold, which I did not use for training. And then I will plot the ROC value of the AUC area under curve value of the held out fold, and I will print it out, and then ultimately I will compute the average of it and find the model that gives me the best average. So here I can show you how it runs step by step. So here I have read the training data, then I have read the test data, and then I have set the loop for the log lambda values that I have to try. Then now I'm importing these libraries liblinear, caret, mlbench, etc. And finally I go to that for loop. When I run the for loop, you will see the graph on the right will change for different values of that penalty, the L1 penalty. So you can see the graph, very high ROC value, and then ROC value is slowly going down as I'm increasing the penalty. And for very high penalty, I eliminate all the variables and I get very bad graph, which is the graph with whose area under that graph is only 1 or only 0.5. Okay. Any questions? Okay. So this is another. Does anyone have
Yeah, you are
question?
audible, sir. There
Okay.
is one question in the chat.
Yes. Let me see. Sir,
Sir,
reference
how?
article for this work. Reference article, I will send it to Smita, and she can share with everyone else.
Sir, how much
Uh,
accuracy you are getting here, sir?
Accuracy for the epithelium versus stoma, I got pretty good accuracy. I think I got around 0.96 AUC. For the other work for predicting cancer recurrence, I got around 0.81 AUC. So that means the accuracy would be around 77, 78%. So obviously that is not good enough for clinical prediction. So we need better models, but we wanted to see how far we can go.
Very good.
So
It's very
yeah, so this other one I will skip. It is a much longer code, but basically it does similar things, fit different models with different penalties and checks them. Okay. So I will pause here and let's take any questions that other questions that people have.
Anybody has any questions?
Hello, sir.
Yes.
I want to know, when do we check precision and recall in a model?
So precision and recall are quite related to sensitivity and specificity.
Okay,
So this area under curve basically looks at both of these together for different threshold values. So it helps us. So if you just look at precision and recall, then you will have to look at precision and recall for different threshold values, right? So any model, if it is trying to classify between 0 and 1, 0 let's say is non-recurrence and 1 is recurrence, any two classes, right? Class A and class B. Uh, Usually it will not just give you a decision of class A and class B, it will actually give you a continuous value between 0 and 1, a probability value in some sense. So the default we think is that if we cut the probability value at zero point 0.5, anything below zero point 0.5 we interpret as 0, and anything above zero point 0.5 we will interpret it as 1. But that is not the only way to think about it. You can actually change the place where you cut it. Okay, so this curve that you get is for different values of the threshold. Okay, so you can take the threshold to be 0.1, 0.2, 0.3, 0.4, not just 0.5, all the way up to let's say 0.9, 0.99, and so forth. And based on that, your precision and recall, there will be a trade-off between them. One will go up, the other will go down.
correct, correct.
Same with sensitivity and specificity. One will go up and the other will go down. So it sort of takes the average of all these models for different thresholds, and you get a single number, which is the area under the curve.
Thank you, sir.
Any other questions? So for any of these machine learning models, such as support vector machines, SVMs, logistic regression, linear regression, for them R is very convenient and it is open source. You don't have to invest money in things such as MATLAB and so forth. You can do it very easily in R. And the beauty of R is that it very easily scales up to very large data. And the documentation of different packages that you can import in R, such as the ones that I showed you, PROC, for example. So if you, I don't know if you're familiar with this, like, so this PROC for plotting that ROC curve, right? If I go to a browser and where is my browser? If I say that PROC package in R, right? We get very good documentation for these packages that tells us who built the thing, and then what are the different uh, uh, commands available in that, how to load the model, how to load the package, and so forth. Um, and also it gives you like some examples of how to use that, how to find confidence intervals of that ROC curve, how to set the sample size, and so forth. So a lot of that documentation is very well documented, and you will see that package after package, the documentation style is the same. Even the documentation style has been standardized. So once you get used to reading the documentation of one package, reading it for another package would be very similar, and you would be very you know where to expect to find things. So this is that online documentation for PROC package. Similarly, you will find for correct package, for lib linear package, and so forth. Any other questions?
Looks like no other questions, sir.
Okay.
They have just started learning R programming.
Okay.
On third day, we are going to give them glimpse of classification and clustering.
Okay, fantastic.
Show them some examples. In that, we'll show them supervised learning, unsupervised learning, and
So, okay.
ROC curve. All that will show them with some small example.
Okay, so this was all supervised learning that I
Yes,
showed you.
Yes.
Okay. All right. Then thank you very much. Smita, if they are, if they want like any references, send me an email. I will send you the references.
Okay.
All
Yeah.
right. Okay.
Thank you, Professor Amit Sethi.
Thank
It
you.
was really good talk, and it was actually really informative. They can see the applications of R, definitely. Yeah.
Okay.
So thank you for guiding us. Thank you.
Okay. Thank you. Take care. Bye.
Bye.